Hello, ladies and gentlemen. This is Alice in Beijing. I'm so glad to meet you again on Friday. So iCAN talks already became one of the phenomena in all this in the whole world for the Society of Science and Technology. Many people would stick with iCAN talks and every Friday. So welcome you come back again this Friday. Yeah, we this week will be a very special week because it's going to be the end of the year. So before we end of the year, so we have one special issue. This is for the young scientists. As everyone knows that we have a Mind Young Scientist Award since 2017. So this was working together with Microengineer and Nanosystem. The journal is one of the top journals in this field. So every year we select several young scientists to be awarded for this title. So actually, we are so lucky to have many of young scientists on this stage for mine. So this time I select three of them. Sheng Xing was win the honor in 2018. And Xu Yang was win the honor and last year, 2019. So Yu Xingge was win the honor this year. So this time we have these three beautiful young scientists, on this stage. So tonight, I think all of you will be enjoy these wonderful talks because the three of them have, very interesting research and then very innovative research on different topics. So without further ado, first I want to introduce the first speaker, Sheng Xing. Xing was from Tsinghua University. He was a graduate there, get a bachelor degree, and then he moved to US to MIT, get his PhD. After 3 years postdoc, and then he come back to China to Tsinghua University to be a tenure track assistant professor. He's doing wonderful, talks on optical electronics devices. His story was started from his implantable devices. Now he's already, get his name on that for this technology. Xing, yeah, we're looking forward to your story. Go ahead, the stage is yours. The word is yours, please.
Hi, hi. Thank you, Professor Zhang, and hello, everyone. Good evening, good morning, or good afternoon. It depends on where you are. And my name is Xing Sheng. I'm from Tsinghua University. First of all, I would like to thank Professor Zhang to provide this wonderful platform for us to get interact with each other and communicate with each other. And we all know this year is it's a very difficult year for everyone. to continue the scientific communication with each other. And so it's my great honor and my pleasure to be here today and share with you about our recent research topic. As I showed here, I'm from the Tsinghua University Department of Electronic Engineering, and also I'm part of the IDG McGowan Institute for Brain Research at Tsinghua University. So basically, my research is sitting in the middle at the interface between the electronic engineering and the brain research, right? And this is the topic I will share with you today. It's about the implantable optoelectronic devices for advanced neural interfaces. So first of all, this is the background, and I want to say something about our brain, right? Human brain usually contains tens to hundreds of billions neuron cells, right? And many more other cells, right? So basically, our brain activity or the function of brain determined by the signal generated within those neurons and their communication with each other. So it's extremely important to develop the techniques to monitor or control the neuron signals. So with that, and we can further understand the complicated structures and functions of the entire brain. With all that knowledge, and hopefully in the future, right, we can develop tools for a lot of useful applications. For example, including the new method to treat the neurological diseases like the Parkinson disease, Alzheimer disease, as such. Right. On the other hand, for people or researchers in the informational technology, right, the understanding of our brain function can help us to design a better computer, right, so-called the next generation neuromorphic computer with a higher intelligence, right? And all of that relies on the development of new technologies. Only if we have new technologies, we can better understand the neuron cells and the brains, right? And help us to develop opportunities for future research and applications, right? This is not only my opinion, right? And here I want to quote from some famous scientist, right? This is a quote from Professor Sidney Brenner, who is a Nobel Prize winner and a very famous neuroscientist. And even him, right, as a neuroscientist, he thinks that the progress in science depends on new technologies, right? New discoveries and new ideas, probably in that order. So he put the new techniques in the first place and the scientific discovery as secondary, right? And also with that background, the United States government launched the program called the Brain Initiative. So it's from several years ago to today, and it's still continuously supporting a lot of new research directions for brain research, especially focus on the innovative neuron technologies to help us to understand the mystery of the brain. Right. So my research interest is focused on the neural interface. Now today, some people call it the brain-machine interface, right? And in Chinese we call 脑机接口, something like that, right? Conventionally, we know that the neural interface is based on the electrical signals because our brain cells continuously generate different electrical signals inside, right? We can use electrical signals to interact with the brain cells and the brain tissues, right? For example, we can use the technique called the DBS, which is stand for deep brain stimulator. We can use the electrical signal to stimulate certain parts of the brain so that we can control the neural activity and restore the functions of some disabled people with the Parkinson disease, right? And on the other hand, we can use electrode to resolve the brain signals. For example, as we showed here, this is the neuron prosthesis, right? And in this case, we use the implanted neuron electrodes to record the signals from the brain of some patients. In this case, the patient is paralyzed, so she cannot control his body because of the damage in the spinal cord, right? But with the aid of this neuron prosthesis, the equipment can directly read out the brain signals, right, from his head, from the brain, and decode those signals and use that to control the external equipment and the robotic motors to help her to finish some tasks for everyday life, right? And more recently, we all know that the Neuralink started by Elon Musk, and he is so ambitious to develop the next generation advanced multi-channel electrodes to interact with the brain, so that hopefully in the future they want to develop these tools for the advanced brain-machine interfaces. Right, this is the conventional method based on the electronics. And on the other hand, right, we know more recently the optics have gradually play more and more important role in the brain science research. This is associated with the genetically development of the genetically encoded actuators or indicators, So this which help us to label the brain cells with specific color, so that we can use a light to interact with those light sensitive proteins expressing in these neuron cells, and can realize very interesting research. For example, we can use optical signal to stimulate and then control the brain signals. Right, this is called the optogenetic stimulation. On the other hand, we can color label the brain cells, different brain cells, and then use the fluorescence imaging technique to detect those brain activities based on the optical signal. So the advantage of the optical signal compared to the conventional electrical signal is that the optical signal has their cell specificity. We can specifically label different brain cells based on their types or different functions, right? So that we can control or monitor the brain signals with higher precision. So here, no matter the conventional electrical interface or the more recent advanced optical interface, right? We all rely on the techniques to interact with the brain, right? For optical interactions, right? The conventional method is using based on the optical fibers, conventionally made by the silica or silicon dioxide. This is the same type of fiber we use for the telecommunication, right? So we use this thin bare fibers implanted in the deep tissue in the brain, in the animal brains, right? And use the fiber as an interface to connect the brain tissue with the external optical instruments like the lasers, photo detectors, and the data acquisition systems, right? So that we can send light in and out from the brain. On the other hand, for electrical signal detection or some chemical signal detection, there are electrodes which have been well established in this area, right? For example, electrodes based on the silicon material or based on the metal wires have been widely used to record the brain signals, right, in terms of the electrical functions or the chemical functions, right? Or use these electrodes to stimulate the different brain regions, right? For those conventional approach, we can see that they're based on these implantable optical fibers or the electrodes, right? They have been widely used. However, they have some limitations at this moment, right? We can see that no matter you use a fiber or electrodes, they are basically a wired system. You need a wire, right? It can be an optical cable or electrical cable to connect the study animals with the some bulky equipment to read out the data, right? It's sometimes created the restrictions for the free movement for those animals, right? So this can be sometimes difficult to study some complicated behaviors. And also those materials we use for the fibers, we use the glass, silica glass. For electrodes, we use silicon or metal. They are basically a rigid and hard material. And this will have a huge mechanical mismatch with the softer brain tissue and create a larger lesion area and more tissue damage. So this implantable fibers in electrodes have not ideal biocompatibility. And also the movement of these cables, right, optical or electrical cables will generate some noise, which will reduce the quality of signal you read. So in our research, because I'm in the electronic engineering department, and my background is about semiconductor optoelectronic devices, right? In this scenario, we would like to design and fabricate those microscale optoelectronic devices. We can incorporate multiple types of the optical devices, right, including the microscale LEDs as light source and the photodetectors as sensors, right, and all the other electrical or chemical sensors, right, to design and fabricate them in the micro or nano scale and integrate all these different components, right, active optical and the electrical components, onto a flexible thin probe made by some biocompatible polymers, and use that as a new optical and electrical interface to interact with the brain. And we can directly insert this multifunctional probe into the brain tissue to replace the conventional optical fibers or electrodes, right? And with that, we can also develop some wireless control circuit to control these devices and read data from them, right? To realize a fully wirelessly operated thin optical electronic probe, and use that as a new interface to interact with the brain. And with that, hopefully, we can get a more accurate and stable neuron signal stimulation or monitoring. Okay, so this is basically all the background of our research, and then I'll talk about the results, right? And in this presentation specifically, I'll focus on the design and the fabrication on some micro scale devices, including those micro LEDs, photodetectors, and filters. And we use these micro devices and we integrate them together to form this wirelessly operated implantable micro probe. And with this probe, we try to address some key questions related to the brain-machine interfaces, right? And because we are focused on the technology part of this, so we try to address some technology difficulties. The first question we need to address that how we can deliver light into the brain, and secondly, we want to solve how to detect light in the brain. And finally, what we want to resolve is that how can we achieve a multifunctions with this thin microprobe? For example, how can we get both electrical signal, optical signal, and even chemical signals? Okay, so I'll introduce 3 different type of microprobes. Okay, the first one I call it the implantable probe for optogenetics. We can use this probe to deliver light into the brain tissue to control the brain activity. And secondly, we developed the probe that can achieve the recording of the brain signals. Especially we use it to record the photometric signal. Basically, it's the fluorescent signals within the brain, so that we can decode the neuron activities. And finally, we develop probe that combine the functions for optics and electrochemistry, so that in this case, we can use this probe to record the chemical signals within the brain. Okay, so let's go to the technology part. So to deliver light into the brain, we need a light source. Conventionally, we use the optical fiber, but as I said, this optical fiber has some limitations, right? Here, we want to use a more efficient light source with a smaller scale to deliver light and directly interact with the brain tissue. Here, we use a microscale light emitting diode. We call it the micro LED. And conventionally, those micro LED, for example, the blue LED is made on a sapphire substrate. It's rigid and hard semiconductor chip, right? And we develop technology to release those thin microscale LED from the sapphire rigid substrate. And then we transfer them onto another substrate. In this case, we can transfer the individual micro LEDs onto a flexible substrate to form our implantable probe. So here shows the freestanding micro LEDs we formed with a size of around 100 micrometers and a thickness only about 5 micrometers. And these LEDs can be printed on a flexible substrate to form the probe we have. And here shows the emission spectrum of the LED. It's in the blue spectrum range, right? And the emission of this LED directly overlap with some light-sensitive proteins. For example, the CHR2 is a classical opsins we developed for optogenetic stimulations. And the LED has a decent quantum efficiency, around 10 to 15%. And here, if we want to make this LED-based microprobe and implant it into the brain tissue, the biocompatibility is extremely important, right? And one of the key aspects for the biocompatibility is their temperature behavior or their thermal behavior, right? And here we can see by monitoring and control the LED operations, right? We choose the correct injection current and the correct pulse width or frequency. We can minimize the temperature effect on this micro LED probes, and we can control the temperature rise for the brain tissue below 1 degree Celsius, so that the implantable LED will not generate extra heat to affect the brain tissue. And so this, after developing those micro LEDs, the based probe, we want to use that to stimulate the neuron signals. Right? This technology is called optogenetics. And here, just a very brief introduction about optogenetics. Basically, optogenetics is a technology that enable us to use light to interact with certain neurons, right? Those neurons already expressed some light-sensitive proteins, so that we can use light with specific wavelengths to control those neuron cells, so that we can use light with different wavelengths to turn on or turn off certain ion channels in the neuron cell, so that we can use that to activate or deactivate the neuron signals. Okay, and the advantage of this optogenetic stimulation compared to the conventional electrical stimulation is that because of this cell-specific expressed processing options, right? We can realize a cell-specific and the color-specific neuron activation. Okay, so this is our technology. We have the micro LEDs, and we integrate them onto a flexible substrate, and then we form this thin implantable probe. And based on this flexible polyimide substrate, the probe has a width about 200 micrometers and the thickness of only tens of micrometers, which has an even smaller scale compared to the conventional silica-based optical fibers. Right? And to control this micro LED, we developed this wireless control circuit to turn on and off these LEDs inserted into the brain. Right, the entire control circuit has a size around 10 to 15 millimeters, and with a weight less than 2 grams, so that they can be used on the animal behavior study using the small mice, the mouse model. Okay, and this wireless circuit can be mounted on the head of the mouse, and without affecting the normal behavior of the animals. Okay, and we have the Bluetooth-based communication technique, so that we can realize long distance communication and wirelessly control the operation of these LEDs with a distance larger than 5 meters. And we have a small battery to power the LED, so that the circuit can last for a long time, with the operation lifetime larger than 2 hours, so that make it sufficient for the specific animal behavior task. Okay, and the key advantage of this wirelessly operated LED probe, as we can see here, is that they will not affect the normal behavior of the animals. Okay, and this is specifically useful and more suitable for study the social related behaviors for multiple animals. As you can see in this case, if we have a optical fiber connected to the animals, and it will restrict the free movement of the animals compared to the wirelessly operated probe, as we can see here. And even worse, if you want to study the behaviors, social interactions of multiple objects, these fibers will entangle with each other, so that make the experiment fail, right? But with our wireless operated probe, so this will not be an issue. Okay, and the next we developed the probe and the wireless circuit. We inserted into the brain tissue and to perform some specific animal task. As we can see in this case, we insert this LED probe into a nuclei called CNF. It's a nuclei in the deep brain of the mouse to control the movement. It's a movement of the animals. As we can see, when we turn on the light to stimulate those neuron cells, the mouse will move and make the circle turns. Okay. So this is just a very simple demo, and we can do a more complicated behavior study, as we show here. Another advantage of using this thin film-based LED probe is that we can integrate multiple different light sources onto this probe. Right? This cannot be achieved using conventional optical fibers, because fiber can only have a single side for light emission. But for this LED probes, we can have array of multiple LEDs, so that when we insert this kind of probe onto a specific region of the animal brains, we can see that different micro LEDs can be targeted on the different area of the brain region, so that we can achieve more complicated behavior studies. We can use different LEDs to interact with the different brain regions independently. Right? As we show here as an example, we insert this LED onto this brain region called superior colliculus SC region. Okay, this can control the defense behavior of this animal. We see that by activating different parts of the brain, that the animal can show different behaviors. Right? As we show here, when this LED is light on, the animal will move very fast, and they try to escape. Right? But when this LED on top is illuminating, the animal will be freeze. It's just so frightened he cannot move. Okay, so that which means that by using this array of LEDs, we can achieve a more complicated behavior study. Okay, and next we can achieve even more functions. Right, we can integrate multiple different devices onto this probe, as we show here, that we can achieve a optical probe for fluorescence sensing. In this case, this probe combines a blue LED, which is used to excite a specific protein called GCAM6. It's a calcium indicator, so that the fluorescence intensity of this GCAM6 will be dependent on the local calcium concentration. Okay, we have another microscale photodetector here, and use that to detect the intensity of fluorescent signals, so that we can use to determine the calcium concentration, and therefore finally we can determine the local neural activities, right? And we have specifically designed the optical filters on top of this detector and the LED to achieve the ideal wavelength selection to get the best signal to noise ratio for specific fluorescence detection. Okay, in this case, we incorporate both micro LED and the micro photodetector together on this thin probe. As we show here, this is the structure of the photodetector is based on a gallium arsenide semiconductor made PN diode. Okay, and we can see that this gallium arsenide photodetector is specifically designed to have a very high sensitivity in the green and the red spectrum range, so that it can have a very high sensitivity to this protein, this green protein, fluorescent protein called GCAM6. And on the other side, this detector has very low detectivity in the blue region, so that it can have the best signal to noise ratio in the brain to exclude most of the blue light, only receive the green fluorescent light. And specifically how we achieve that, we design a method to integrate the microscale optical filters on these devices. As we show here, the conventional optical filter usually based on the multi-layered oxide material. As in this case, showed it's made by multi-layered silicon dioxide and titanium dioxide. Right, this high performance, high quality filter usually are deposited on the rigid substrates like glass or silicon. And in this case, we develop a method to release those thin film filters from the original rigid silicon substrate and transfer them onto our microscale devices. As we show here, we can transfer a filter on the micro LEDs to tune the LED emission spectrum so that can only let the short wavelength light pass through without passing the long wavelength light. And on the other hand, we can transfer a band pass filter on top of the photodetector so that we can shape the detection spectrum of the photodetectors. As we see here, the band selective detector will restrict the detection range of the diode within a specific range so that it can achieve high signal to noise ratios, only sensitive to the green and the red fluorescent light. Okay, and similarly, we developed this wirelessly operated circuit to control this photometric probe, so that we use a battery-powered circuit so that to control and turn on the LEDs, and we use an amplifier and the micro CPU to read out the photodetector, the current signal from the photodetectors, and we use the infrared signal transfer method to wirelessly transfer the photo signals to the external data acquisition system. And these devices, it can be implanted into the different targeted brain regions to read out the fluorescent signals in freely moving animals. Right? As we show here, the key advantage of this probe is that it's based on a thin film material, so that the lesion area created by this probe is even smaller than the conventional optical fibers, which usually has a cylinder shape and create a larger damage on the brain tissue. Okay, so that this flexible substrate-based probe had better biocompatibility. And in terms of the signal recording, as we show here, we use some stimuli to stimulate the animals. As we can see, we introduce an electrical shock to the brain, to the animals, and it will induce the fluorescent signal change within this specific target brain area. As we show here, that we compare the signal readout from the fiber optics based equipment and our wireless device. We can see that both systems can achieve a comparable signal to noise ratio. And finally, I want to introduce another technique. We can do optical stimulation and optical detection, but we can even do more, right? Because our implantable probe, this flexible probe, provide a platform to integrate all kinds of different sensors on top of it. As we show here, we introduce a optoelectrochemical probe to combine the functions for optical stimulation and chemical analysis. As we show here, specifically, we vertically stack different devices together with a LED in the bottom and the diamond in the middle and the P-dot PSS layer on the top. And this LED will provide light for optical genetic stimulation, similar to our previous work, right? We can use light to stimulate the neural activity. And this P-dot PSS layer is a conductive polymer, and it has very high chemical sensitivities. We can use it to do the electrochemical sensing. Specifically here, we can use it to sense the signals of some neurotransmitters, for example, the dopamine inside the brain. And the diamond layers here show it serves in between the LED and the P-dot PSS. It offers optically transparent and thermally conductive interface to isolate the P-dot PSS layer and the LED layer. And that we show here that the key component in this optoelectrochemical probe is this diamond interface. As we show here, we know that diamond is very hard, rigid material, and it's also optically transparent, so that it can let the LED light to pass through without blocking the light. And also it has the highest thermal conductivity, so that it can be used to reduce the temperature rise associated with the LED operation. And finally, this diamond interlayer can also be a waterproof layer, so that it can protect the LED from the moisture or the tissue, the liquid, for introduce the damage. Right? As we show here, that this diamond interface is extremely important as an interface layer in between the P.PSS layer and the LED light source. And the P.PSS on top of diamond provide a very good tool for the highly sensitive electrochemical detection. As we show here, that this P.PSS layer can have a very high sensitivity on the dopamine signals. And we first of all tested those the device performance in the aqueous solutions. We dropped the liquids with different dopamine concentrations onto the P.PSS layer. We can see that this P.PSS chemical sensor can detect the change of the dopamine signals. The detection limit is around 0.1 micromole, and it can be used for to analyze the dopamine levels in the animal brain. And finally, we also insert this microprobe into the specific region of the animal brains. And also similarly, we design a wireless control circuit to power the LEDs and read out the electrical signals from the P.PSS layer. As we show here, we insert this probe into a specific region called VTA. It's in the deep region of the mouse brain, and it can have it's basically this VTA is a resource for the dopamine release. Okay, as we see here, that when we turn on the LED, and the LED will stimulate the VTA region and increase the dopamine level. As we can see that this will make the mouse happy in the left side, right? When the LED off, then the mouse will be less happy. So that the mouse will like to hang out in the left part of this behavior box, okay, because the light is always on in this case. Okay, at the same time, we can use this P.PSS based chemical sensor to sense the dopamine level change, as we show here, that it can be used to detect the electrochemicals with a high sensitivity within the brain. Okay, with that, here I think I want to wrap up. In this presentation, I showed you how we utilize those microscale optoelectronic tools, how we design and fabricate those microscaled optical devices, including micro LEDs, photodetectors, and the filters, and we incorporate them all together into a thin, flexible platform to make this integrated optoelectro and chemical probe. And we use that to wirelessly control or monitor the brain activities. Specifically, we address those three important questions. We use this probe to deliver light into the animal brain to control the brain activities. And also we use this probe to receive or sense the light signals inside the brain to record the brain activities. And finally, we combine the different functions, including optical function, electrical and chemical functions, into this probe, and use that to simultaneously stimulate the brain activity and read out the chemical signals inside the brain. Okay, and I think I have 1 or 2 minutes left, and I'll briefly introduce some other directions are going on. And as we show that this optical brain interface is a very new direction, and there are a lot of work we can do, right? The thin probe we formed can be multifunction. We can incorporate all kinds of different devices and sensors on top of it, and use that to interact with the neuron system. Okay, and I just show you a few examples. As we show here, we can make LEDs. We can make the light source with different emission spectrum. As we show here, our semiconductor design allow us to fabricate the micro LEDs with different emission wavelengths that cover the entire visible spectrum, from the violet to blue to green to yellow and red. Okay, And we can even fabricate array of those devices. As we show here, we can use that to display different colors, right, and show different color patterns. And some display companies are very interested in this, and they want to use it to make the next generation display system. Okay, but our interest is more on the biomedical applications. As we show here, the reason we develop those different light source is that we can use them to target different light sensitive proteins, so that we can use this different light source, different color, to independently control those different proteins, so that we can use them to interact with different neuron cells. Okay, that's our goal. As we show here, we can implement multiple light source all together in a same probe. As we show here, this is a multicolor LED probe. It can display different colors, and different color can be used to interact with different proteins. As we show here, we express two different proteins in those neuron cells, and we use two different colors to control them. When you shine the blue light, it will inhibit the brain signals, and when we shine the red light, we will use to activate the brain signals, so that we can realize this, we call it bidirectional neuron activation, neuron signal activation and the inhibition at the same time. Okay, and we can also develop new technologies to transfer the energy. In the previous work I showed you, I used the battery to power the LED inside the brain. Okay, because battery is very stable and it can work for a long time. The disadvantage of a battery is, it's so heavy, right? It's big. It cannot be fabricated in a micro scale. Okay, in this case, in this work, we develop new technologies to harvest light, and we develop a device, micro scale device. They call it the optoelectronic copper conversion device. In this device, this device can receive the infrared light and convert the infrared light to visible light. And we know that the biological tissue has a very good transparency, high transparency in the infrared part, so that when we insert those microscale device into the brain tissue, and we can directly use the infrared illumination, external infrared illumination, to activate those micro LEDs inside the brain and achieve very high luminescence, visible light generation inside the brain. Okay, and finally, we develop new materials. We can develop more biocompatible and biodegradable materials for the brain-machine interfaces. In this work, we develop new types of optical fibers based on the biodegradable material called the PLLA, polylactic acid. It's a biocompatible material, and we can use that to make an optical fiber with very good transparency. We can use that to deliver light, and it's more flexible. And more importantly, this fiber can degrade inside the tissue. As we show here, when we implant the fiber in the brain tissue, and this fiber will naturally dissolve inside the brain. So after 2 months, and we can use that potentially for some clinical applications. So in this work recently published, we use this biodegradable fiber to regenerate the damaged bone tissues inside the animal body, right? And after the operation, this fiber will naturally dissolve inside the body and make it ideal choice for the clinical application. Okay, and finally, I think I will end here. I'll wrap up. First of all, I would like to thank all of my group members, including my students and my postdoc, who work very hard and develop all the techniques we use for the biomedical applications. And all the techniques I show here today are based on their efforts. And also, I would like to thank our collaborators, mostly at the Tsinghua University and the Peking University, and at the Chinese Institute for Brain Research in Beijing, and also collaborators from Shenzhen Institute for Advanced Technology. And finally, we are hiring, if you are students or postdocs and you're looking for new positions, and we are interested. And no matter what background you have, no matter you are an electrical engineer or optical engineer or material science or even biomedical engineer, and you can find a good fit in our team. Okay, and last but not least, and I want to thank you for your attention.
Okay, great, Xing. Yeah, very nice
Thank you.
talk. A lot of information. Actually, we have a few questions here. The first question is you're using PI. For how about the mechanical property and the long-term stability of this material? Can you give some comments on this? This was from audience here.
Oh yeah, the PI is the full name is called Polyimide. It's a very standard material people have been used for a long time for the flexible electronics and for wearable sensors. People use it to attach to the human skin and make those epidermal sensors. They are based on the polyimide. Okay, it's flexible and it's I think the modulus and the stiffness is orders of magnitude lower compared to the conventional optical fibers, which is made on the glass. Okay, but still maybe it's rigid. It's harder than the brain tissue, so that we can insert it into the tissue, right? And some people are trying to develop a even softer material. Okay, but if the material is too soft, for example, gel-based material, and it has better biocompatibility, but the difficulty in that case is how can you insert the soft material into the brain? It's so soft, it cannot stand alone, right? You have to use some other method to attach to a rigid substrate to finish the surgery and then release the rigid substrate, something like that. And in terms of the stability, I think the polyimide substrate has a very good stability in the biological environment. And we even have this PI-based micro probe with LEDs, and we have a good waterproof encapsulations on top of the LED, and we insert the probe into the animal brain, and it can last for more than a year.
Oh, really?
Yeah.
That's
Yeah.
pretty long.
Yeah.
Okay, very good. Yeah, then for the people who want to use polyimide for these implantable applications, now you should have confidence because she already did a wonderful job at using this material. Okay, the next question is, yeah, I think in your last part of your talk already answered this one. Yeah, Professor Xing, very impressive work. Talk. Please go back to the questions. And did you try any other different colors LED? Yeah, so you
Yeah,
already.
I think I briefly introduced the work. It's not published yet. We mostly use the blue LED to stimulate the ion channel called the CHR2. It's a very standard, well-established opsin, which can be well expressed in the different type of neuron cells. So that we choose blue LED to stimulate the CHR2 expressing cells, right? But if we want to achieve a more diverse the functions, so if we want to achieve the stimulation and the inhibition at the same time, we have to introduce more different options. And this another option should have different excitation spectrum compared to CHR2. So that's why we need separate options, right, with different sensitivity at different wavelengths. And in that case, we'll have a different light source. Basically, we need different LEDs to stimulate those different options. Okay.
Okay. Very good. Actually, your last part already talk about this.
Right.
Yeah, you already done a lot of work on that. We're looking forward. Here I have one question is following this. For optogenetics, a lot of people was working on this, and now you already done something. Can you give some words for what's the future about this, field, optical genetics?
Oh yeah, I think optogenetics is a very great success in terms of the biological science, right? It's a tool developed, I think, more than a decade ago, and people now use it in different areas. It's a very powerful tool for brain science research, so that we can use the different options to label different part of the brain or different cell types, and so that we can use the light to interact with them. So that's the cell specificity and selectivity is the key advantage compared to the conventional electrical stimulation. So that it can be a very good tool, a powerful tool for the scientific research. In terms of the the clinical or medical applications in the future, I think right now, based on what I have known, that the optogenetics have been approved for the retinal repair, or that for the patients or people with the with some defects in the retina, they cannot see things because the retina cell is damaged, right? So that they can now scientists are trying to use the optogenetic method to
repair that.
to express the light sensitive proteins back into the retina cells so that make them sensitive to light again. Okay. That's
Very
one.
good. Actually, you may listen to Ed Boyden's talk on iCAN X. That's a few weeks ago. Yeah, it's inventor of this field. It was putting a lot of efforts on that. I'll be very glad to know you already done so many things here.
Yeah, well, we're trying to develop the new tools to facilitate the research in the optical neural interfaces.
Okay, very good. Yeah, the last question is pretty long. Professor Xing, thanks for your wonderful talk. As shown in your talk, the device is a little bit bigger for implant applications. It may cause a problem for animal study. What's a big challenge for you to shrink the size and also the power supply?
Right. In terms of the device, we have two components, right? The probe is very small. It's even, as we show, is even smaller than the conventional fibers. Okay, it has better biocompatibility. In terms of the circuits module is relatively big at this moment because I'm mostly a device guy. I'm not an expert in the circuit design, but we have good collaborators in our department who can design the better circuit for us, and we can use the customized IC chips to incorporate all the different functions in a small chip, so that we can shrink the size of the control module, right? But in terms of the power supply, another issue is the battery we use. Actually, the battery we use dominate half of the weight of the circuit. Okay, so there are other methods have been developed. For example, my postdoc advisor, Professor John Rogers, and he had even started up a company to commercialize those things in his technology, and they used this RF circuit based magnetic coupling method to transfer the power to the micro LEDs, so that it's basically a battery free, so they don't use a battery in their circuit. Okay, that will reduce a lot of burden.
Yeah, I think the next few years you may develop your own ASIC, for your devices, and all these power issues and the size issues will be, get over. Yeah.
Right. That will be a very good research direction in the future.
Yeah. Thank you very much, Xing. As iCAN X tradition, we provide you this electrical version for your wonderful contribution to iCAN X. So we're proud of you, the new technology and all the job you done. It was really using the high tech to connect the world and the universe. Thank you very much.
Thank you, Professor Zhang. Yeah.
Okay, thanks.